After nucleoli, second part that is present in nuclear matrix is the chromatin. Chromatin is loose or indistinct network. We can also say that it is highly extended or elaborate network. Network is of nucleoproteins. So here nucleoprotein fibers will be present, and these fibers are best seen when the nucleus is in interphase. This particular chromatin will be present in interphase, but it will be converted into chromosome, it will be condensed into chromosome in different parts in different phases of cell division. This particular chromatin or the chromosome contains DNA. There will be basic proteins. Basic proteins are called as histones. There also will be some non-histone proteins. RNA will also be present in this chromatin or chromosomes. Talking about the length of chromatin, in human cells, the length is approximately 2 meter, and this 2 meter thread condenses into 46 chromosomes, and when these condense, then this chromatin is known by the name of chromosomes. In this particular video, we will look into the detail of chromosomes, the structure which is present. As we very well know that chromatin is the thread which when condensed converts into the chromosome. When we talk about the structure of chromosome, we talk of a point that is called as primary constriction. So, this is the structure of chromosome. Here, this particular point, the middle point where these chromatids are attached, this particular is technically known as primary constriction. Its second name is centromere. So, we can say that primary constriction or the centromere, it will hold the chromatids of chromosome together. Along with this centromere, one more structure is present, that is called as kinetochore. Kinetochore is shown as greenish structures in it. Kinetochores are disc shaped structures, and these will be attached to this particular centromere. As we will study further in cell division, spindle fibers during cell division attach to these structures called as kinetochores. After this, coming to the types of chromosomes, here we will talk about the 4 types of chromosomes, and it will be based on the position of centromere or the primary constriction. The first type is called as metacentric. Second type is called as submetacentric, third is acrocentric, and the fourth is telocentric. The difference lies in the position of centromere. Let us try to form these structures. The first chromosome that we talked about was metacentric. In metacentric, centromere will be exactly at the middle point, and the chromatid size will be equal, and that is why we call it metacentric or V-shaped chromosomes. The second type is submetacentric. So, coming to second type, submetacentric. In this, centromere will slightly be away from the center. So, if this centromere is slightly away from center, so one chromatid will be a bit smaller and second chromatid will be a bit larger, forming a L-shaped, and the structure is called as submetacentric. Moving to the next one, that is called as acrocentric. In acrocentric chromosome, the centromere will be towards the end. So, next is acrocentric. So, in this it will be very close towards the end. So, this is the centromere. So, very small one arm will be there, and the second arm will be quite long or large, and this is the case of acrocentric, and this is also called as J-shaped chromosome. The last chromosome is telocentric. In telocentric chromosome, centromere will be located towards the terminal end or just towards the end, and that is how we get telocentric chromosome. So these are the 4 types of chromosomes depending on the position of centromere. After looking into the types of chromosomes, we come to the term which is called as satellite. Initially, we had talked about the structure primary constriction or centromere. If second constriction is present, then that is called as secondary constriction. So let us see in this particular diagram, or we can make over here. So this is the primary constriction that we talked about. If one more constriction is present, say for example, one constriction is present over here, let us form it by green color, and this part is present. So this second constriction is called as secondary constriction, and whatever part is left here, this part is known by the name of satellite. So satellite will be the fragment which will be present after secondary constriction. There are certain important features about this satellite. Satellite is non-staining. The point to be remembered is that chromatin was staining, but satellite will be non-staining. It will always be present as a constant location, and it gives appearance of a small fragment as we had seen in the diagram, and this was only called as satellite. We can also see the description that we have covered in these particular diagrams. Here the arms are equal, so this is the metacentric. In this, the one is short arm and one is the long arm. So these both diagrams are of submetacentric, and in this one arm is very small and the other arm is quite large. So this is the acrocentric chromosome.